In the first section, we talk about the brief history of cell biology, and then we move into the development of the cell theory. Then we talk about the different cellular dimensions, sizes, and shapes of cells. Now we're going to talk about how we're able to observe these different sizes and shapes, and we're going to focus on microscopy. We're going to talk about three different types of microscopy. One that is the different variations of light microscopy, that in this case we use light to and different optical tools to see the different samples. Then we will talk about other tools, yeah, like staining, like the microtome. Then we will move into another type of microscopy that we are not using the same type of um, uh, white light. We're going to use fluorescent light, and we're going to talk about, okay, what is fluorescent? And then we're going to talk about different microscopes that use fluorescent. There are many. Here we're just going to talk about the uh, white fluorescent microscopy and the confocal microscopy. Then we are going to briefly talk about the electron microscopy. That while in the first one we were using white light, visible light, in the second one we were using this process of fluorescence. In the last one, we have a much higher resolution because we are not using light or fluorescent. We're using electrons to make those pictures, okay? So they give us a much higher resolution. Okay, so let's start. Okay, so there are different type of microscopy. The first one that we are going to study here is light microscopy. That is the first one that cytologists use as a tool. Right now we have different type of microscopy, and here you can see a picture. And I want that you take a notes. You can stop this video and try to think about okay, could you see in the bright field? Do you see something? Do you see what is the problem? What do you notice about this one? Question, is this two-dimension or is a three-dimension picture? Okay, what is the difference between this one phase contrast and this one dark field? Okay, what is the difference in the contrast and the polarized light? Okay, what is happening here? So I want that you observe those one. In all of this one, what we are doing is we are using different tools of physics, yeah, to change the light in different ways, to use all these principal light optical principles to create different brightness and contrast of the samples. Okay, so here what we are doing is that in the bright field is wonderful to study different structures. However, there are different cells that if they're alive, they can be transparent. Yeah, yeah. So what we need is to play. If we want to study them, we need to stain them and kill them, or we need to use different optical tools. So one optical tools is this one that is very common that maybe if you are in a healthcare profession, you might use is the DIC microscope. That this one you use optical tools will play with the light to create a 3D picture. Yeah, it's not actually 3D. It's like this is a square, and if I play with perspective, with yeah, I this look like a very ugly cube, but it gives you the impression that it could be three-dimension. Here we're playing with light to give the idea that there are structures that are above or ones that are below, yeah, and this is all of this we do it playing with optical with physical principles. Okay, this one is called phase contrast. This one we are actually going to use in the lab, and it's pretty cool. Yeah, so here again we're playing with, if you remember, light are waves. So what we're going to do is to play with the refraction of the light. So the things that are different brightness here inside of the cell and here outside of the cell. So in that way we have more contrast. Well, here everything look in the bright field. Here there's little contrast between the cell and the background. Here in the dark field we have much more contrast between the cell and the background. Something similar is happened here in the Hoffman modulation contrast. We're not going to study this one in a lot of detail, but again, we're playing with physics to do that. In the dark field and in the polarized light, we're doing something similar, but in this case, we're making the field, the background dark, and yeah, we're making the structure bright, okay? And here we just play with the light to make it of a different color. Okay, so in these cases, what I was showing you is that different type of microscopy that allow us to study cells without staining them and to be able to study it without modifying the cells. Okay, so this light microscopy allows us to study nuclei, mitochondria, chloroplasts within the cells. It allows us to count number of cells. It's very useful to study live cells in some cases. And what we're doing is called bright field microscopy because white light is passed directly through the specimen. Later, we will study other type of microscopy where we use different type of lights, like fluorescent lights. Yeah, so we are going to study that in the following slides. So as I said before, and you will see in your textbook, we are going to uh, study in the lab different microscope, and the microscopes will have generally these four components. You have your objectives, yeah, and both, for example, in the bright field and in the dark field, both will have objective. We will have the specimen here in your slide. We will have here a light source. Here you have a light source that in this case is bright field. In our microscope, it could be fluorescent light that it could make, it will give us, you will play with different rules. And here, for example, you can play with different optical tools to change the light. And in this case, in the bright field, as I said, you see the cells, the background is white, and the cells in the border look dark, while in the dark field is the opposite. The outline of the cells here in the dark field is white, while the background is black. Okay, so the dark field, for example, it will not tell you a lot of information what is happening inside of the cell, but it will be very good to outline the cell. Okay, so again, each type of microscopy is useful for to answer different type of questions, and in the lab we will talk more about that during the semester. So now we're going to talk about another improvement in microscopy, and this is not related to a different type of microscopy. It's related to how to make the samples. So this tool is called a microtome. A microtome, micro is small, yeah. The root of small in Greek, and tome is about cutting. So this is a device that allows to make very thin slices. And here you can see an example of a microtome. So here you have your samples and you have something that allow you to cut it very thin. So if you remember, in your introductory biology, you had you had to cut a thin slice of an onion that is wonderful because it has large cell and is very easy to cut. However, samples that are very difficult to cut. So for example, I have to cut fly legs that are very thin and difficult to cut. Those type of structures, those type of samples it will be easier with a machine. So a microtome is used to make a slice, yeah, to preparing very thin, several micrometer of a slice and it allowed to do it rapidly and efficiently. Yeah, so it helps you to do it very well. Of course, as any tool, it have some limitations. Yeah, that so as we talked in the previous type of microscopy, the light microscopy, you can do some things, but you cannot do other ones. So one of the limitation is that you are only going to have a small resolution of different of very few micrometers. Yeah, you cannot make thousands of slices. Yeah, it will be a waste of time. To do that, you will use a different type of microscopy that we will talk about that later. Okay, so this one is a very good tool, the microtome. It allows to make very thin slices, and it have uh, some benefits and some disadvantage, but it's a wonderful tool to use. Now let's talk about another improvement in microscopy. So in addition to the microtome, another way to improve the microscopy is to create a variety of dyes. And these dyes are used to stain the cells and that allow you to create a better contrast with the surroundings. Yeah, so there are different type of stainings. So maybe you have seen this one, very clear. That is for gram staining. So you can see bacteria that, and you can see some of those bacteria. In this class, we will use this one, the methylene blue, to see the viability of the cells. Very useful. So there are many different type of stainings, and why we use it, because if you remember, there are cells that are in the bright field that are transparent. We cannot see them, so we can use different tools, or we can just stain them in different ways and to create for color them, and in that way it's easier to differentiate what is a cell and what is an artifact, what is a debris, or we can identify, for example, create a staining for a specific protein that is in the membrane. So we stain only this area of the cell, or we can see in the lab, for example, that we can have stainings for the only here in green will be the nucleus, or we can have for the actin. So it's a very useful tool. Okay, so we have bright field microscopy, we have microtome, we have stainings, and now we're going to talk about the other biggest step, that is to create specialized light microscopes. That right now um, this is that one of the things that we use the most. So now we're going to move into these very specialized microscopes that are sometimes very expensive. Like some of them are $500,000 or even more expensive. And I know that most of you are not very interested in this topic in detail. Some of you are are going to study ecology, or ones are going to get into healthcare profession. But if you are interested in cell biology, you can talk to me. There are courses online for free that talk a lot in detail about this. So I'm just going to give you very general ideas, and if you want to know more about all this type of microscopy, there is a lot of videos available that I would love to share with you. And those videos are, for example, some of those are Nobel Prize winners, some of them are the key uh, scientists that developed that technique, or that are the pioneers on those new techniques. So with that introduction, um, we have a variety of special optical techniques that have been developed for observing live cells. You might see some of my movies, and we might analyze some of the movies if you're interested. And some of those techniques include the phase contrast microscope. So we are going to use that one in the lab. We are also going to talk about this fluorescent microscopy. My movies are used this one, that is the confocal microscopy, and this one that we talked in the past, that is the DIC, differential interference contrast microscopy. This is the one that give you images that look like 3D. Yeah, the confocal microscopy will also give you pictures that look like 3D, but using fluorescence. Okay, so let me talk about this in the following slides, and over the semester we will talk in more detail about those ones. Okay, so now I'm going to talk a little bit more about the phase contrast. And here again, bright field, amazing, has helped us a lot. However, there are times that when you have living samples, that if you see this picture is much easier. If I ask you, please count the cells here, you say, Nicholas, this is too difficult. you will hate me. However, in a phase contrast, life will be much easier, and you say, this is clearly a cell, this is clearly another cell, this is a, I don't know, here there is a piece of garbage, a debris of the cell, so maybe we shouldn't count that. So this is very useful. Same thing happened with the DIC that create 3D pictures. If you want to know more about that, again, here is the one of the word expert. And the idea is that this technique cannot be used alone. So for example, you should compare this one to this one with other techniques. So you know that they, again, each one has their own limitations. So what I told you is that there are different techniques like the phase contrast and the DIC, differential interference contrast microscopy, and those one are very good to see live cells like this one. Why? Because they allow you to use the transmitted light, yeah, and create that these the low density samples. You can enhance and amplify those slight changes. And how? Because you're playing with physics. In this case, you create um, different density between the cell and the surrounding. And later we'll get into the physics. But the idea, the magic there is that you are creating that those small changes in brightness become much higher, and you can see much clearer the samples. Okay, so again, I told you about this phase contrast and the AC, very important tools. Now I'm going to move into another tool that is the fluorescent microscopy that we are going to also see it in the lab. Okay, so we finished with this section about light microscopy. And now we're going to move into the fluorescent microscopy. And to start, we're going to ask the question about fluorescence. What do you think that fluorescent is? So in the following slides, I'm going to talk about fluorescent. Right now, I just want that you see two different sources of fluorescent. One here, these people in a party, yeah, that for some reason, we don't know why, you see that their clothes are fluorescent, yeah. And here in the following slide, I will show you that here these clothes that these people have are absorbing wavelength, in this case the UV range, and they are also emitting. There's an emission of this fluorescent light that appear in those clothes. Yeah, something similar is happening in the biological world in a process called that you have seen maybe before, you have heard about documentaries, it's just beautiful. It's the process of bioluminescence. And here in the ocean, you can see, if you have seen that, it's just beautiful how different organisms in the ocean all, both produce and emit light, and that has a huge importance in cell biology because we were able to use that power of bioluminescence to label specific parts of the cells. Okay, and we're going to talk about that in a second. To start talking about this process, I'm going to show you a video. This is the video. If you want to watch it here, again, here is the link, and I'm going to show you this fantastic phenomena that is the bioluminescence, and they will talk about how humans were able to use the bioluminescence for cell biology.
Imagine a place so dark you can't see the nose on your face. Eyes open or closed, it's all the same, because the sun never shines there. Up ahead, you see a light. When you creep in to investigate, a blue light flits around you. I could watch this forever, you think. But you can't, because the mouth of an anglerfish has just sprung open and eaten you alive. You are just one of many creatures at the bottom of the ocean who learned too late to appreciate the power of bioluminescence. Bioluminescence refers to the ability of certain living things to create light. The human body can make stuff like earwax and toenails. But these organisms can turn parts of their body into glow sticks. It's like nature made them ready to rave. Why? In one way or another, bioluminescence improves a living thing's chances of survival. Take the firefly. Its ability to glow green helps it attract a mate on a warm summer night. But it's just one of many living things that can glow. The railroad worm, Phrixothrix hirtus, can light up its body in two colors, red and green. Would you eat something that looks like an airport runway? Neither would any sensible predator. The flashing lights keep the worm safe. Then there's the deep-sea shrimp, Acanthophyra purpurea. When it feels threatened, it spews a cloud of glowing goo from its mouth. Who doesn't run the other way when they've just been puked on? Plus, that puke attracts bigger predators who want to eat the shrimp's enemy. So what if you can't bioluminesce? No problem. There are other ways for living things to make bioluminescence work for them, even if they weren't born with the equipment to glow. Let's revisit the anglerfish moments before it tried to eat you. That glowing bait on top of its head? It comes from a pocket of skin called the esca. The esca holds bioluminescent bacteria. The anglerfish can't glow there by itself, so it holds a sac of glowing bacteria instead. Remember the firefly? It can actually make itself glow. Inside its lantern are two chemicals, a luciferin and a luciferase. When firefly luciferase and luciferin mix together in the presence of oxygen and fuel for the cell called ATP, the chemical reaction gives off energy in the form of light. Once scientists figured out how the firefly creates its luciferase and luciferin, they used genetic engineering to make this light-producing reaction occur inside other living things that can't glow. For example, they inserted the genes, or instructions, for a cell to create firefly luciferase and luciferin into a tobacco plant. Once there, the tobacco plant followed the instructions slipped into its DNA and lit up like a Christmas tree. The beauty of bioluminescence, unlike the light from the sun or an incandescent bulb, is that it's not hot. It takes place in a range of temperatures that don't burn a living thing. And unlike a glow stick which fades out as the chemicals inside get used up, bioluminescent reactions use replenishable resources. That's one reason engineers are trying to develop bioluminescent trees. Just think, if planted on the side of highways, they could light the way using only oxygen and other freely available clean resources to run. Talk about survival advantage. That could help our planet live longer. Do you find yourself thinking of other ways to put bioluminescence to good use? That glow stick you swing at a rave may help you find a mate, but how else can bioluminescence improve your survival? If you start thinking in this way, you have seen the light.
Okay, so let's recap. So I hope that you enjoyed the video, and now I want to remind you what is fluorescent. So in the video they show you that fluorescent is the emission of light. So in the beginning I show you people with different clothes that they were fluorescent. I show you in the video this bioluminescence, different organisms that are fluorescent, and what happened at the chemical level. So here you need to understand a little bit of energy, and here you have this compound. Okay, so what is happening, let's put it in green here, is that for some reason a chemical start becoming fluorescent and start emitting a light. This is what we see. So we see that this is emitting a light, and what happened here at the energy level is that actually is not producing high energy, it's actually emitting low energy. So here you see the process here. When you see that something is fluorescent, the electrons here are going from a high energy to a low energy, that it was unstable. Good. And why that happened is because before that there was no different process, that they were absorbing light. So that chemical, yeah, the one here, this molecule before emitting that light have to absorb light that have high energy, so increase in energy, and then to go back into the normal to the ground state, it will emit light and go back. Yeah, so we go from a high energy state to a lower energy state. Okay, and here on purpose I put two type of colors. I put one blue and one green, yeah, because in the microscope we will see that. Later in the lab you will see that in the microscope you will see a laser that is blue, and when you go to the eyepieces you will see a completely different color, that it will be that the sample turn green. So it absorbs blue light, high energy, and it emit green light, that it will be a lower energy. Okay, now I told you there were these eye biology lectures that they are great. Now I'm going to show you 10 minutes, or no, 10 minutes, no, 2 minutes of a lecture that they do those experiments, and you will see how they take chemicals. They have a laser and they of one color, and the chemicals that they will show you there, different chemicals, produce light of different colors. Okay, key process again, emission, so absorption that lead to emission. High energy state to a lower energy state.
Water, pour it in a glass, and then the other part is that we need UV light to illuminate it, and that you can buy in the form of what's called a urine tracker in the pet store. And so when we now shine this UV light in the quinine, in the tonic water, and you see that you get a blue light emanating from within the tonic water. It's another wavelength. It's a longer wavelength than this light with which I illuminate. And that's why we see it a lot better. Our eyes are not very sensitive to this UV, but they are highly sensitive to the blue light in the tonic water. Quinine is not the only compound that fluoresces, of course, otherwise it would be quite uninteresting. Already in the 19th century, scientists synthesized this dye called fluorescein that is highly fluorescent. And when I shine blue light in that, we get yellow fluorescence coming out. And another very well-known dye is rhodamine. And when you shine green light in that, you get this orangey, yellowish fluorescence coming out. And so this is fluorescence. Okay, so what we have seen.
Great, I hope that you enjoyed that section, that experiment. And I remind you, there are 4 steps in the fluorescence, in the process of the production of fluorescence. So the first thing that he showed you is that when you use a laser, you have the absorption of energy. So here, all of these molecules go and absorb the light, and when they absorb the light, these create an increase in the energy level. That is, this energy level is unstable, so it goes to an excited state, but this state is unstable, so it has to go back. And how it goes back? By emitting light here. So it emits light here, and this emission of light create a wavelength shape. Yeah, so from a blue in this case that is a high energy stage to a one that has a lower energy that is a green state. Okay, so 2 key sections. In the beginning, an electron is excited by a high energy photon, and then there is a relaxation of that electron that causes the emission of a photon of a lower energy that the one caused the excitation. So it goes from blue to green light. And as they show you in the video, it doesn't always have to go from blue to green. It can go from different colors. Yeah, and I think that was very clear. Okay, so yeah, I hope that you enjoyed that experiment, and if you want to know more about fluorescence, this is a very important expert on the field of fluorescence. Here is the entire video lecture that they talk a lot about in detail about this process. I don't want to overwhelm you with so much information, but I want to tell you one key component of, okay, now we know what is fluorescent. Now why we use that? Why we use? Because it produces a high contrast. So you remember that we were talking about phase contrast, dark field. Here, this one it produces also high contrast. But in addition to that, it produces high specificity. Yeah, here you can say, okay, clearly the background is here in black. Yeah, so you can clearly see that this is not a part of the cell. But you can also see that in the cytoplasm, you have something green. So here, let's put a C here. This will be the cytoplasm, and here the nucleus will be here. Yeah, and in the nucleus you can see, for example, the chromosomes. Yeah, so it can allow you to see in more detail what is happening and be sure of what is happening here. So one are the microtubules in the cytoplasm. The other ones could be the ones that are going to move the chromosomes. Yeah, so not only contrast, they also give you specificity. And I'm going to show you in a second why we can have such a high specificity. Although in the video they show you why, because you can have or probes, or you can include the fluorescent in a specific part of the genome. So you can say to the microtubules, add them and make them fluorescent, and the chromosomes, proteins in the chromosomes, in the histones, to become fluorescent. One green fluorescent, GFP, the other one RFP, red fluorescent protein. So it gives us both high contrast and high specificity. Also, you can have you can quantify those. Yes, you can say, okay, look, definitely there is these things here are green. Yeah, these microtubules here are green, but they are definitely more concentrated than the ones that are in the cytoplasm. Okay, so there's a higher density of the molecule there. What this means? Yeah, so you can start quantifying those things. You can also create a mutation, for example, in the microtubules, so you will not see a lot of that. So when there's low microtubules, maybe you cannot separate the chromosomes. So it can allow you to create these quantifications. And we are going to cover some of those things later during the semester, how to quantify all these pictures and how to study this process. Finally, you can also do live imaging, and you have seen hundreds of my movies, maybe multiple times. So I use fluorescent to do all those kind of things. So you can not only see the sample dead, but you can also see them alive and see all the process of, for example, this one will be the cell division. Okay, now that we know what is fluorescence, now I can show you how fluorescent microscopy work. And to do that, I have this great video that I found that it was very useful for me to understand that. So in the beginning here, they will explain, compare what is the transmitted light microscopy. So here, how they illuminate from here, that it goes here and go through the sample, and later you end up seeing that using your eyes here in the eyepiece. And on the other hand, in the fluorescent microscope, you need the laser that goes here with, in this case, blue light, that have a mirror that goes and illuminate the sample, excite the fluorophore that is there, and then as a result of that, it goes with a different wavelength here, and you can see it with the eyepieces. Okay, so uh, hopefully this video is clear, and then I will go through the key steps that they will be in the lab.
In this animation, we will introduce you to fluorescence microscopy. Fluorescence microscopy is a special type of light microscopy in which the microscope uses a laser as the light source. A researcher will require the use of a fluorescent microscope to visualize autofluorescent cellular structures, fluorescent dyes or stains, or to analyze the expression, modification, and/or localization of proteins using fluorescent antibodies by a technique called immunofluorescence. In a fluorescence microscope, the light source travels in a different trajectory than in the basic light microscope that we illustrated in the microscopy experimental technique animation. Instead of the laser light being transmitted through the sample, as in a simple bright field-based light microscope, the fluorescence microscope uses incident or reflected light. From the laser light source, the excitation laser beam travels through the filter cube, which is used to provide the appropriate wavelength of light to excite the fluorescent molecules, or fluorophores, used in the sample, then through the objective to focus the laser light on the sample. The light emitted from the sample is then reflected back through the objective and the filter cube, which filters the emitted light to the camera or eyepiece where the researcher can view the fluorescent signal. One advantage to fluorescence microscopy is that it can be used to detect and visualize multiple fluorescent molecules by simply adjusting the excitation and emission filters to detect the relevant fluorophores in a sample. Fluorescent microscopy also has many useful applications in the laboratory setting. Fluorescent dyes or stains are used to detect cellular structures. One example of a commonly used fluorescent dye is DAPI, which binds to DNA, and a second example is phalloidin, which binds to F-actin. Expression, modification, and/or localization of specific proteins can also be visualized using immunofluorescence, or IF. Like immunohistochemistry, IF uses multiple steps to detect specific proteins using antibodies in the sample. In IF, one of the antibody labeling steps utilizes fluorescently tagged antibodies that are detectable upon excitation using the appropriate wavelength of light.
Okay, so let's recap all the things that we have learned in this slide. You learn what is fluorescent. You know the importance of the absorption and the emission of light. Now you know how to apply that principle into a microscope, and you saw that a microscope generally can have the equipment for transmitted light, and in that way you locate the sample, but at the same time then you can use the fluorescent microscopy, and this is what how our microscope work. Generally, when you go in the microscopes in the lab, when we are going to use the fluorescent microscope, for example, we are going to use the legs of the flies, you will find it first with the transmitted light, with the light microscopy, then you can see it, you can turn on the fluorescence to see how they fluorescent. Okay, so those both work at the same time. So very important. Now, key thing to remember about the fluorescent microscopy is that it allows you the detection of specific proteins. So I show you a picture, if you remember, of a cell that have the chromosomes in red and the microtubules in green. And how we do that? Because we know that the process of microscopy allow you to have one protein that you can excite in blue, yeah, and then you can see it. It will emit a light in green, and you can see in that way the microtubules. Yeah, and you can also have a different protein for for the um, the chromosomes. Yeah, so what we have in the second, so this can, let's put this too for the, and let's put an M right here, an aim for microtubules. But you said I also want to see the chromosome, so you can label the histone, you can excite the histone with a green laser, and then this will emit um, a red color. Yeah, and here you're able to see also seed of the chromosomes, and you can keep adding and adding more and more protein. So remember, it always go from high energy to low energy. So high energy here to low energy here, high energy here to low energy here. Okay, the other thing that we talk about, and I am sure that you remember it in the video, is that you can put fluorescent proteins, yeah, in the DNA, and this is how we do it. I'm going to show you later, if you haven't seen some of my movies, that we do that. So we're able to put that at the DNA level, and we can also make stainings of that. And in the next slide, I will show you how those stainings work. Finally, the last thing that I wanted to show you is the different steps. And in the video, you already see it. So just to remember, beginning, what happened here is that you have a light source. This light source in the fluorescent microscope is not going to be white light, it's going to be here is going to be a laser with a specific light. In this case, the emission light will be blue, yeah, and it has to go to a filter, so it doesn't go everywhere. filter the light, and this light is going to bounce to the sample and is going to produce, it's going to excite the electrons in the sample, and those one are going to get excited, go to a higher level of energy, and then they're going to, when they go back into the ground state, they will have a shift in the light, in the wave light, and they produce a green light. Let's put it here in green. And this is going to go to an emission filter that is going to filter all this light and is going to go to a camera, and then you can see that. Okay, so I hope that all this thing is clear. If not, you can watch the video again. And now I'm going to show you how these fluorescent probes work. So now let's think about how we do an antibody staining. Yeah, so you can have antibody staining with fluorescent molecules. So what we need three things. One thing, an antibody. Another thing, a fluorescent molecule. And another thing that you have is an antigen. So three things. The antibody, yeah, it can be a protein that is located in the cell. It can be located, for example, here in outlining the membrane. Yeah, you remember cell members have protein, that protein is located there. Now what you need is to be able to attach this to that protein, to attach the fluorescent molecule to that antibody. So how you do it? You chemically, you play with chemistry, and you have the antigen, and you create a link between those two. And when you create those links, then you put it in the sample, in the cell, and the antigen is always going to attach to the antibody. So as a result, these three molecules will be together inside of the cell. The, the fluorescent molecule is going to already be attached to the antigen, and the antigen, when it finds that specific complementary antibody, they are going to attach. Okay, so what I told you is that antibodies are proteins, yeah, and they bind to a particular target molecule, and those target molecule will be an antigen. Okay, so that we take advantage of these phenomena that we know that happen in all organisms, and we can target specific proteins. And then what we do is that the antibody can be coupled with a fluorescent molecule, and in that way, now we can outline any specific part of the cell. Yeah, then we can go and attach it and see specifically if you want to outline the cell, if you want to outline the microtubules, the chromosome, etc. Okay, so now that we talk about fluorescent microscopy, now we're going to talk about, this is called, this type of fluorescent microscopy has also divided in different fields and different type of microscopies, and I want that you observe these two type of pictures, the one here and the one here. Yeah, and if you see, if you compare the one here to the one here, are the same, but one of them have better resolution, the other one looks blurry. Same thing happened here, yeah, and specifically here. If you see here, this look like with a higher resolution, look much better. So one of those, the one, the number one, the one on the top, are uh, are taken with a white fluorescent scope that it illuminates the entire sample, and the second one is using a fluorescent microscope that allow you to remove all these excess of fluorescent. Yeah, it's like when you see the sun. If you look at the sun, not a good idea. Yeah, it's too bright. You need specific glasses, or you need a telescope that remove this excess of fluorescence. So what you see, or when you turn on a light and you look at the light, yeah, it's too fluorescent. To see it better, you need to reduce the excess of light. So what we're going to do is to have a microscope that filter the excess of light, remove them, and we can have more even more resolution. So again, this is a relatively complicated topic. So I'm going to show you a video that compare both the fluorescent scopes that we have been talking, the one on the top that illuminate the entire sample, to the more the one that give you better resolution. That is the confocal scopes that illuminate only a specific section of the sample. Okay, so let me show you the video in a second. So this video doesn't have audio, so I'm going to explain what is happening there, but I think it's very clear. So here, if you want, you can read what they said here, and what they are showing you is the different components of this microscope. Um, you know, the different filter, and here the laser, and in a fluorescent scope, you have a large uh, laser that is illuminating here these three cells. So it's illuminating, and there is a lot of light. This produces the process of fluorescence. So now they are absorbing the light and they are emitting the light as we talked before. And this is what you are going to observe in the eyepieces or in a computer, that generally those when now they have attached a computer. Okay, so this picture is not going to be it's going to be relatively blurry, have a high contrast, but it's going to illuminate the entire sample. A confocal microscope is slightly different. Yeah, so although it will have a laser, it will have different filters and different pinholes that if you see, you don't illuminate the entire sample, you illuminate only a section. You're not illuminating the three cells, you're illuminating only a specific section of the sample. And this is going to create a filter, is going to produce, sorry, the emission, and is going to also have another pinhole that is going to only illuminate, that is going to show you the light for that specific section. However, now you need to start scanning the other section of the microscope. So one cell, the second cell, and you have to go very slowly. While the other picture, you just have one picture. Here, you are going in 3 dimensions. You're going to go in the x-axis, in the y-axis, and also in the z-axis. So it's very useful, it's very pretty, but while the other picture will take 10 seconds, this one can take minutes, 2 minutes to take, so and or even longer. So it's very useful. It will give you not a 2-dimension picture, it will give you a 3-dimensional picture, but it has benefits and it has disadvantages. Better resolution, but this picture will take much longer to take. So here I just recap all the information about confocal microscopy. One of the advantages of confocal microscopy from the wide fluorescent microscopy is that here, as you have much more clear samples with less blur, these one are much better to quantify what is happening. So you can see the spatial and temporal patterns of the distribution of the protein, so you can clearly see. In the other one, you see that the chromosomes are separated, but with the confocal microscope, you can see much clearer in which direction they are going. You can quantify them, you can see that reduction in the fluorescence. In the other one, there will be just too much light. So this one is better for quantification. Another thing that is important is that in the confocal microscopy, remember, you are not illuminating the entire sample. Yeah, you are using a laser to illuminate a single plane. And how you do that? You use different pinholes. Yeah, so not all the light is going there. Just going to is going to be light for a single place. Yeah, in the sample, and then again is going to so you're going to use this mirror to transfer the light to the sample, and the light that is going to emit also is going to have a pinhole, so it's not saturated, and there is just the correct amount of light to see the sample. Finally, as I said before, here you are not going to be, this picture will take a long time. you're going to have to scan one section in the x-axis, so it's going one direction, then it's going to go in the other direction. You can see this can take minutes, and then you can go also in the z-axis. So you need to store those pictures, yeah. So they will have a camera and have a computer that we will collect all the pictures, and then you need a software that we will learn later in the class to stack all those pictures to see all these dynamics that you will see in videos like this one if you want to watch it. Okay, so now to finish, I'm going to show you the last video, and we are almost done. Okay, so now you're an expert on these two types of microscopy, and now we're going to move into the last one, that is the electron microscopy, that have has the best resolution so far. And I'm going to talk about two: the scanning microscopy and the transmission microscopy. So to that, again, this video is going to show you that this type of microscopy has such a good resolution that is not only used in cell biology, but is used in our fields of biology or in our fields of science. So let's start with that.
Have you ever wondered how scientists and engineers design transistors that are around the width of a strand of DNA? How do we even take pictures of such nanoscopic transistors? Well, that's the role of the electron microscope, which has literally changed the way humanity sees the micro and nanoscopic world. Don't believe us? Take this European peacock butterfly, for example. When we zoom in on its wing using a light microscope, we see that it's composed of tiny overlapping scales. But when we zoom in using an electron microscope, we can clearly see the shape of each scale, and zooming in further, we see how the scales have a truly incredible texture entirely foreign to anything that humans manufacture. Although this wing may not be directly related to the technology you're familiar with, scientists and engineers have been using electron microscopes for the past 60 years to develop smaller and smaller transistors. And with today's technology, this microscope can zoom in millions of times to where it's able to capture images of individual atoms. There are two main types of electron microscopes. The scanning electron microscope, or SEM, is used to see surface images like this butterfly wing, or the bristles of a used toothbrush. See, here are cells from your body, and all around here in yellow is bacteria. It's gross, but let's move on. Scanning electron microscopes have a maximum resolution of around 1 nanometer, meaning the spacing between two adjacent features or dots of resolvable data in an image is 1 nanometer. The other type is the transmission electron microscope, or TEM, which is used to take images of structures that are inside materials, much like an X-ray machine takes pictures of the bones inside our bodies. For example, TEMs are used to take the pictures of these sections of a transistor. However, in other domains of science, TEMs can be used to take images of proteins inside mitochondria, the powerhouse of the cell, or of nanoparticles of pure gold. Transmission electron microscopes are typically more complex than SEMs and have a resolution up to 50 picometers, which is roughly the size of a hydrogen atom.
Okay, so now I hope that you enjoyed that video. I'm going to give you key points so you remember for the test. So first, the electron microscopy is divided into different type of microscopy. The first one is the SEM, and the second one is the TEM. And the SEM is the scanning electron microscopy, while the TM is the transmission electron microscopy, and both use an electron beam. We are not using light, white light or fluorescent light. Here we use electrons, and we use it in different ways. In this case, the SEM, as we watch in the video, and with the butterflies, they are used to study surface samples, while the TEM allow you to study internal structure. So both can be used in different ways. The SEM will give you 3D pictures, while the TEM will give you only two-dimensional pictures. The magnification is different. The, the SEM has a lower magnification, and this is ridiculous, 2 million magnification, while the TEM will give you this 50 million of magnifications. As a result, the resolution of the SEM could be 0.4 nanometers, while the other one will be 0.5 angstrom. So very powerful microscopes, and they give you amazing pictures. However, different from the fluorescent scopes, yeah, you cannot have colors, yeah, the colors that we will show you in the pictures. You have to make them, so they should be, you use different software to create those colors. So in other words, it could be very useful. It can give you a lot of resolution, but again, as usually all the microscopes, they have advantage and disadvantage. And here the super advantage of this is that it can give you a huge resolution. Okay, so again, we're going to watch just 2 more minutes of this video that explain very well how this microscope work.
Behind a TEM is that it generates electrons and accelerates them to around 70% the speed of light, thus creating a beam of electrons. Next, a series of magnetic lenses focuses the electrons down to a small area and shoots or transmits these electrons through the specimen that we're looking at. Depending on the different densities and materials inside the specimen, the electrons are scattered as they pass through it, thereby imprinting an image of what's inside the specimen onto the beam of electrons. The imprinted beam of electrons is then magnified 40 times using an objective lens, and further magnified 50,000 times using a set of projector lenses. At this point, the imprinted image is 5 or so centimeters wide and large enough to be captured by a high-resolution camera sensor at the bottom of the microscope. We'll explore the detailed engineering in a little bit, but for now, you might be wondering, why do we have to go through the hassle of manipulating electrons, and why can't we just use light? Well, visible light is physically limited to magnifications up to around 2,000 times, and if you try to zoom in further, the image remains blurred without revealing any more details. On the other hand, electrons can reach meaningful magnifications up to 2 million times. Why then is light physically limited? Well, let's return to this image of the European peacock butterfly and the scales on its wings. This image was captured with a camera. This image was taken with a light microscope. And these images were captured with an electron microscope. Let's consider two features from the specimen that are only 100 nanometers apart. Visible light has an average wavelength of 540 nanometers, which is larger than the distance between these 2 points. Due to the physics of waves, as light hits these two features, it's bent around, thus creating a pair of propagating waves with a diffraction pattern resulting from the interference of the two waves. If the features are substantially closer than the wavelength of visible light, then the diffraction pattern will make the two features appear like a single blurred feature. In short, visible light can't really resolve features that are less than 300 nanometers apart.
Okay, so again, I think that video was amazing and simplified things a lot. If you want to know more about it, I left the link there. They talk even in more detail, even beyond to what we want to know in this course. But here, the key idea that they give you this wonderful resolution. As I said before, unfortunately, you don't have colors. You create this color with a software that it make the pictures easier for you. When you know very well your sample, you can help other people to understand better what they are supposed to be observing, like here and here. Yeah. So later in the course, I will show you how there are multiple software to do that. And here, if you want to know more about the electron microscope, you can go in your textbook. However, key information here is that here you're using a beam of electrons rather than light, and now we know that the light has some problems with the resolution. As we watch in the video, with the electron we can have even higher resolution, and so it allowed that these microscopes are 100 times better than the light microscopes, and you can have a magnification that is wonderful, like up to 100 times power. That is incredible. So you can see, as they describe in the previous videos, like study how the proteins are folding and all these type of things that are fantastic to study. For example, how the function of the protein. So you can see, for example, many videos of simulations of how the um, hydrogen pumps in the cells work. And one way to do that they got these pictures was using this type of microscopy, and later they use computer simulations and animations to show us actually how we think that they work. And this would have been impossible only with the confocal microscopes or type of microscopy. Okay, so just to finish, I told you that there are two types of microscopy: the transmission electron microscopy, that in this case, the one that you see here, if you see there are two dimensions, the electrons are transmitted through the specimen. If you want to know more about that, in the video there is a lot of more information. I didn't want it to overwhelm you with more physics about it, but they explain that in that video. And in the scanning microscopy, that you can see here, this allow you to see the surface of the specimen that is a scan, and how they do it by detecting electrons that are deflected from the outer surface. So as a result of that, in one microscopy type of microscopy, you can see things in 3D. Yeah. Well, in the other one, you only will see the pictures with much more resolution, but only in 2D. Okay, so with this, we finished all this information about microscopy. I hope that this excites you, and we can see now the exam and try this in the lab. And I hope that you will enjoy this a lot. Of course, there will be more information in your textbook that I recommend you that you read. Questions of the for the test will include, I will show you pictures like this ones, and you have to tell me, okay, which one is bright field, which one is confocal microscopy, what do you think that is fluorescent microscopy, what is phase contrast. Yeah, and hopefully I could have a practice test so you can practice, but a good way to start practicing is to read the textbook and see this type of pictures. Okay, I hope that you enjoyed the class, and if you have any questions, just let me know. Bye.